Thank you. It's a pleasure to be here and to talk a little bit about my work on the microbiome and the preterm infant. When you think about the microbiome and preterm infants and socioeconomic status, there's a couple of ways that one can think about it. Certainly we know that the incidence of preterm delivery is associated with socioeconomic status. There's a lot of work that the microbiome is changing during the course of pregnancy and that the microbiome itself may trigger delivery. So it would be wonderful if someone could understand that and solve that so that there were no more preterm babies. But unfortunately, we're not there yet. So I am not looking at the causes of prematurity. So certainly those are associated with the microbiome and socioeconomic status. I'm taking it from the time that the baby after birth arrives in the neonatal intensive care unit. And my goal is sort of understanding how we can optimize conditions for that individual baby so that they can thrive and have the best possible outcome. So just starting with some context and some definitions. What is the definition of full term? People know? 37 to 40 is this current strict definition. So the definition of preterm? Anything under 37. There you go. Limit of viability currently. 22 to 23. Possibly 21 if you're in Japan. But there's questions about their dating. So we will routinely resuscitate down to 23 weeks. There's always a conversation with the parent, 22 weeks if they are really insistent that we attempt everything. Below that, we really do not currently have very good success. And so we are generally consider the limit of viability 22 weeks. So I think these pictures sort of help demonstrate what I am sort of working with. So in contrast to this healthy full-term baby born at 40 weeks gestational age that everyone is hoping for, this is a preterm infant that I could easily find in my nursery, born at just 23 weeks gestation. So you can see next to the parent hand that this baby is small. But that's not actually the primary issue. The primary issue is immaturity. So I think you can also see from this picture that the skin is immature. You can see that the baby's eyes are still fused, that the baby has a breathing tube because the lungs are immature. And although you can't see it from the outside, the intestine and brain are immature as well. The smallest survivor in the Guinness Book of World Records is still a twin born at 8.6 ounces. For context, there's your 12-ounce Coke. And that baby was born at Loyola, and that baby is doing well. But in general, what do we know about survival for these infants? So extremely low birth weight infant is defined as a baby born at less than 1,000 grams. And it's probably not a surprise to you that the smaller you are, the lower the survival. So at birth, there is a birth weight dependent survival curve. But what do we know about the survival? And this is data from my colleague Bill Meadow here at the University of Chicago. If you look at the day of death for the non-survivors, the majority of them die on day 1. Actually, the vast majority of them will die by day 4. So that if you actually look at the survival for patients who are alive on day 4, so the patients who are able to live to day 5, the birth weight specific mortality disappears. So for every day that you are in the neonatal intensive care unit, the odds increase that you will survive and go home, which is exactly opposite what you see for the adult intensive care unit, where every day that you're in the intensive care unit actually decreases the odds that you will survive and go home. So that's a completely different economic discussion. But what this says to me is that there are a lot of survivors. I think people worry a lot about the survival of preterm infants. But survival is actually pretty good, even for the smallest, even for the 500-gram baby. What I think our challenge is what does long-term outcome look like for these babies? And I think it's important to remember that there really isn't anything wrong with these babies except that they were born too soon. So the issue for them is a change in environment before they were expecting it. So if you think of a preterm baby as essentially a 3rd trimester fetus, expecting the sterile intrauterine environment, an environment where the placenta does all the work of providing nutrition and oxygenation, this missed time change in environment is very significant for these babies. Preterm infants account for about 12% of births, but they disproportionately account for about 40% of children who have cerebral palsy, 25% of children with hearing impairment, 35% of those with vision impairment, and certainly many others with mild or moderate neurodevelopmental difficulties. They find themselves in an unexpected environment, the neonatal intensive care unit. In this environment, there's medications, there's delayed feeding, breast milk versus formula, H2 blockers, opioids, instrumentation, exposure to plastic. So this is where I sort of suggest my interest is the preterm infant population itself, but I really think that the preterm infant is sort of a window for all of early development. So if you think of the preterm infant as representing the third trimester, exposure to stress, plastics, separation from mother, some of these other things can actually be looked at in the preterm infant. And this is an article that just came out in the last couple of weeks discussing the value of the preterm infant as sort of the canary in the coal mine. And what they say is, like the canary in the coal mine or asthmatics in air pollution studies, children born preterm may serve as a sentinel population owing to increased susceptibility to the sometimes modest effects of common intoxicants, improving study power and decreasing necessary sample size. So again, my interest is the preterm infant, but I think a lot of what we can learn from this patient population is actually relevant to other infants as well. So how did I get started on the microbiome and the preterm infant? It actually began with a clinical question for me, and this is where a lot of the studies on the microbiome and preterm infants began, with a look at the disease neonatal necrotizing enterocolitis. So this is an inflammatory bowel disease that primarily afflicts premature infants after the initiation of enteral feeding. This is just the definition. You can see that from this, there's no indication that we understand the pathophysiology or can predict what babies will get this disease or have any treatment, because that's really the status of where we're at. What we know is that there are several risk factors. Prematurity itself is the primary risk factor, along with bacterial colonization, also enteral feeding, and hypoxia or altered intestinal blood flow. Babies who have this disease have significant mortality in the nursery, but they also have significant morbidity long-term. So there was a study of several infants looking at neurologic outcomes of these babies. It was a multi-center retrospective analysis in the late 90s. Infants in the neonatal network, less than 1,000 grams. Over 5,000 were entered into the registry. Almost 3,000 were evaluated at 18 and 22 months, and there was even split between those who had necrotizing enterocolitis that required surgery to actually resect part of the intestine, and those who could be managed medically, that means with just antibiotics and bowel rest. And what they found was those who had the most severe necrotizing enterocolitis, so surgical neck, had increased risk of cerebral palsy, a mental developmental index less than 70, physical developmental index less than 70, or any neurodevelopmental impairment. Surgical neck was specifically associated with periventricular leukomalacia, so that's white matter injury for the babies, bronchopulmonary dysplasia, so lung inflammation, again cerebral palsy, and decreased growth for all parameters. So I'd just like you to hold on to that connection between growth and neurodevelopment. We'll come back to that. But this is sort of an indication that the short-term morbidities that we see in the nursery have long-term implications as well, beyond just survival. So we initially put forward the hypothesis that enteral feeding, putting together all the risk factors, enteral feeding results in colonization of the uniquely susceptible premature intestine with pathogenic bacteria, resulting in an exaggerated inflammatory response. And we originally thought that the issue would be pathogenic bacteria, that we were looking at this as a baby who was developing well, and then there was an insult, and that if we could identify this pathogenic bacteria, we would have something to treat. Culture studies had not revealed anything, but it was really hoped that the microbiome technology that we had newly available to us would finally give us the answer. So we asked the question, can microbiome analysis be used to identify the pathogenic bacteria associated with NEC? We had few patients, just 20 patients. We had 10 with NEC, 10 control, but we did have 4 sets of twins in which one had NEC and one did not. And then we just looked at the fecal samples prior to the onset of NEC by 16S sequencing. So it's as I mentioned, we don't have a way of predicting what babies will get this disease. So we have a biorepository where we have fecal samples of many preterm infants collected from birth. And then when we have an outcome of interest, we can look retrospectively at those samples. So we paired babies who had NEC with a baby who did not develop NEC and matched them by mode of delivery, gestational age, and day of life. And just looking at diversity, many have spoken about diversity as something that is commonly associated with health. You can see that the patients who went on to develop NEC had lower diversity than the control patients. And just at the phylum level, whereas control patients had about 40% proteobacteria, the majority was other phyla. Of note, these control patients really are our healthy patients. 40% would be considered very abnormal in any adult population. But in the patients who go on to develop NEC, there's a bloom in that proteobacteria increased to almost 100% of their population. The other phyla become less than 10%. But if you then look at individual patients, the top chart is the patients who develop NEC. The bottom is the control infants, and they're paired. So N1 through N4 are the first NEC patients, and the C1 to C4 is their matching twins. And then the others were unrelated matches. What is labeled is the dominant genus, and you can see there is no single organism that causes necrotizing enterocolitis. However, for the top NEC patients, each of those labels is a gamma proteobacteria. So the NEC patients did have a bloom of a gamma proteobacteria to more than 50% of the community, which is not something that we saw in the control patients, except for these two patients here. C9 and CN10 were chosen as controls. At the time they were chosen, they were healthy, met all of our criteria, but interestingly, they actually both went on to develop NEC 7 and 10 days later. And when we looked at clustering, we saw distinct clustering. In the blue squares are the control patients. The red dots are the NEC patients. And you can see separation, except those two patients, again, clustered with the NEC patients. So this was sort of our first indication that although we couldn't identify a specific organism associated with NEC, that there was perhaps a community associated with NEC, and that the change occurred before disease. But these were only single time point studies. So we decided to look at more longitudinal samples, and we looked at shift over time. So if you look at the initial samples, these were for the twins. The initial samples are pretty close together. The pie charts just represent phyla again. But those who will go on to develop NEC follow one trajectory, whereas those who remain healthy follow a different trajectory. And the difference seemed to be associated with actually the decrease in the firmicutes, which changes the balance. This change occurred several weeks before the onset of NEC, so again indicating that early life events are perhaps setting something up that leads to disease later on. And at this point, we sort of had a decision to make. We could either follow the patients who went on to develop NEC to try to understand more about disease, or we could follow the patients who remained healthy to try to understand a little bit more about what kept them healthy. And that's what we chose to do, to follow the healthy patients. And so we took those patients and looked at their samples over time. We compared those in the samples from preterm infants to those of a healthy full-term breastfed infant. Those are the black squares, and that has generally been considered the gold standard of health, that preterm infants should always be compared to this full-term infant. But we found for those patients who didn't develop NEC, those who remained healthy, there was actually a temporal progression even for them. Their samples didn't follow the same pattern as the full-term infant. There was a clustering for our preterm infants in the first 2 weeks of life, another clustering between 3 and 5 weeks of life, and then the open squares are samples past 6 weeks of life, and you start to see that those then begin to finally converge on the pattern of the full-term breastfed infant. The samples from the breastfed infant are also over the same time frame, over the first sort of 8 weeks of life, and this is a baby cared for at home. So while there is variation, those patient communities are pretty similar to each other, and you can see that for the preterm infants, there is a temporal progression. To bring this back to NEC and sort of the idea of how important are these really early time points, there have actually been only 3 clinical interventions that have been shown to have an effect on the incidence of NEC. Probiotics, so obviously that's an overt manipulation of the microbiome. But 2 others that didn't actually reference the microbiome are days of antibiotics and breastfeeding. So these were large studies that just looked at a clinical intervention and incidence of NEC. And interestingly, each of them are interventions that modify the microbiome in the first 2 weeks of life. So if you consider your intervention giving probiotics, there's a decrease in NEC. If you consider your intervention withholding antibiotics, that decreases your incidence of NEC. Or if you consider your intervention giving any breast milk, that decreases your incidence of NEC. And that study on breastfeeding was specifically breast milk in the first 14 days of life. So there seems to be common themes that this first 2 weeks of life is important. And when we thought about it, one of the key differences for the preterm infant population compared to other populations is really this active development of the infant while they're in the nursery. So a baby will stay with us for many weeks to months. What we tell the parents is it takes 40 weeks to grow a baby, whether that's in the mom or in the nursery. There are some time elements of development that really just have to go through the requisite time. So for our patient populations, host development coincides with microbiome development. And so the samples from the first 2 weeks of life are really the earliest microbiota that sort of encounter a naive intestine and really have the greatest opportunity for affecting fundamental pathways. And so we decided to look at how does that earliest community influence health and development, and needed to ask the question, well, how do you measure that? There's actually not a great definition of health, and development can mean a number of different things as well. But this is a paper that came out in 2006 that just looked at growth. So growth in the neonatal intensive care unit influences neurodevelopment and later outcomes for extremely low birth weight infants. And the conclusion of this manuscript was that growth velocity during an ELBW infant's NICU hospitalization exerts a significant and possibly independent effect on neurodevelopmental and growth outcomes at 18 to 22 months corrected age. So we took growth as our marker of health. Growth represents sufficient nutrition, resistance to infection, resilience during their course in the nursery. For those of us who are in the nursery, we know there's always these babies. It doesn't matter how many calories you pour into them, they just don't grow. And growth really is the most important job for a preterm infant. It determines when they can go home. It determines when they have enough sort of fat on them so that they can maintain their temperature and not have to be in an incubator. So we took 2 babies who had very different growth trajectories. You can see high growth in the red, and we define that as greater than 10 grams per kilo per day growth. The poor growth or low growth in the blue was less than 10 grams per kilo per day growth. We sequenced them, and from here going forward, H represents high growth, L represents low growth. We took samples from 12 days and 6 days of life, so again, the first 2 weeks of life. And what you can see here is what our initial sequencing showed. You can see, first of all, there's actually not that many organisms. So this would be very low numbers compared to other studies in older children or adults. There are some areas of overlap, but also some distinctions between the 2 groups. And what we did is we used a mouse model. So we took the fecal lysate from the infant, transphonated that into a germ-free mouse, and allowed the pregnant mouse to deliver her litter so that the pups acquired the microbiome from the mother in sort of a natural fashion through the birth process, through being with the mother, through nursing, and then we followed the growth of the pups. And what we found was the fecal sample from the high-growth baby led to high-growth mouse pups, and the fecal sample from a low-growth human baby led to low-growth mouse pups. So we were able to recapitulate the phenotype just by transferring the microbiome. But now we have the opportunity to look at the tissue in ways that obviously we can't in babies. So the first thing we did was we looked at the intestinal tissue, and it really doesn't matter what all of these letters and genes represent. You can see low growth and high growth. These are gene networks associated with the inflammatory response. Red or the orange color indicates upregulation. And the most important thing to notice from this is that there's a lot of orange in the low growth and not so much in the high growth, indicating that there was upregulation of genes associated with inflammation in the low growth group just from transferring the microbiome. There was no secondary inflammatory response. These pups were not sick. The only difference was they had a difference in their growth trajectory and had received a different microbiome. When we, by PCR, looked at specific genes, again, at the intestinal level, blue is the low growth, the magenta color is the high growth, and you can see, again, upregulation of genes associated with inflammation, IL-1 beta, TNF, MCP1, in the low growth compared to the high growth. But it didn't just stay there. When we looked at the serum of these mice, we see the same pattern. So again, blue is low growth, the magenta is high growth, compared to germ-free or SPF, which would just be considered normal mouse colonization. And at every point, the low-growth microbiome led to increased inflammatory markers in the serum in addition to the intestine. We then looked at some degree of mechanism. We looked at NF-kappa B activation. So NF-kappa B is a transcription factor associated with upregulation of inflammatory and immune-associated genes. The brown color indicates positive staining, and we again compared low growth and high growth to germ-free and SPF. And what you can see is consistent with the other data. So low growth has upregulation of NF-kappa B, but interestingly, the high growth actually had lower levels than even SPF. So compared to sort of normal colonization for a mouse, the high growth was actually able to decrease inflammation. And this was important to us because in previous studies, we had shown that a key difference between immature and mature intestine was upregulation of this NF-kappa B pathway, leading to an exaggerated inflammatory response. So this was an indication that, again, just the earliest microbial colonization could affect these pathways in either a negative or actually a positive way. And thus carried over to the cytokine level. So when we look for staining of specific cytokines, low growth versus high growth, low growth has lots of staining, lots of the inflammatory response, high growth has less. So why does this matter? This matters because for our preterm infants, actually most of the morbidity of prematurity is associated with inflammation. It's not just intestinal inflammation, necrotizing enterocolitis, but also lung inflammation, bronchopulmonary dysplasia, inflammation of the eye, retinopathy of prematurity, or of the brain, periventricular leukomalacia. These are the primary morbidities associated with development in the nursery and perhaps a consequence of what microbial initial colonization these babies receive. And how does this tie to long-term outcomes? Well, if you look at all of these morbidities, there's 4 of them here. Add another 2. So this list has 6 morbidities in addition to the 4 I mentioned, adding another one, inflammation associated with sepsis or intraventricular hemorrhage. If you just count the number of morbidities, 0 to 6, that is specifically associated with neurodevelopmental impairment at age 2. So the more morbidities you have, it doesn't matter which ones they are, the higher the likelihood is that you will have neurodevelopmental impairment at age 2. So you start to see how your initial preterm course in the NICU begins to have long-term effects on these babies and what they're able to do. So we wondered if all of this kind of feeds into neurodevelopment, does the microbiome affect the brain as well? So we took our same mice and we looked at the brain tissue. You see low L and H once again. If you recall, for both the neck and the paper on growth, growth is associated with neurodevelopment. This is a staining for neurons. So you can see much higher staining in the high growth compared to low growth. And we also did a stain for myelin. So that affects sort of how messages get sent. High growth has more staining for myelin than the low growth. So it suggests, again, that just this early basic colonization affects brain development at the cellular level. We decided then to look at mechanism to try to probe a little more deeply, and we decided to look at IGF-1. So we have established that certain microbiota normalize growth. And mutations in IGF-1 are found to be associated with both growth failure and neurologic effects, microcephaly, and developmental delay. In rodents, IGF-1 results in reduced brain size. We know that germ-free mice have lower circulating levels of IGF-1, and IGF-1 crosses the blood-brain barrier. So this seemed like a reasonable target, and this was all work done by Jing Liu, who is a research scientist in my lab. She looked at the hypothesis that microbial colonization can modulate brain development through regulation of IGF-1, and started off by looking just at serum levels. So if you look at serum IGF-1 levels, there's your low growth and high growth again. High growth has higher levels at both 2 and 4 weeks. And these are significant time points because 2 weeks is prior to weaning, 4 weeks is post-weaning. And then she looked at the brain levels. So in the top 2 panels at 2 weeks and 4 weeks, there's not much difference between low and high at 2 weeks, but by 4 weeks, the high growth has higher levels of brain IGF. But if you look at the bottom panels, it's not based on transcripts. So our thought is that this is actually IGF-1 coming from the serum. Looking a little bit closer at receptor levels, there isn't a difference in receptor, but IGF-BP1 is a binding protein for IGF. It prevents the IGF from interacting with the receptor, and the high growth actually has lower levels of the binding protein. So it suggests that the microbiome is affecting serum levels, which transfer to the brain, and then affects how well it is able to affect the brain. So up until now, we've been talking about neurodevelopment in the hospital and the effect of the microbiome there. But our goal is not just that, again, it's not just that these babies survive their NICU course, but that they thrive long term. And traditionally, the follow-up for these babies has been at age 2. And there are standardized tests that you can do to measure neurodevelopment at that age. But what we are interested in is sort of long term and looking at school readiness as a metric, because school readiness is very functional. And that does feed into if you are able to be in a regular class at age 5, what does that mean sort of for your long-term life as we think about socioeconomic status and opportunity. And this is a study by Mike Massal here at the University of Chicago, along with a fellow, where he took the babies who had a certain outcome at age 2, they're on the x-axis, and looked at what their development was at age 5 when measured by school readiness. And what he found was that there was a big difference. So even if you were normal at age 2, there was potentially more than a 25% likelihood that you were not ready for school at age 5. And the biggest determinant of this was not your hospital course in the nursery, but rather the socioeconomic status of the family that you go home to. So the difference between these bars is Hollingshead Index of Social Position. So this just includes maternal education and occupation, but you can see there's a difference both in the normal and the slightly delayed group, that the variation post-NICU is really based on the home that the baby goes to. So if we take our curve, it suggests that there's something that shifts this curve one way or the other. And we focus all of our energy on optimizing the time in the nursery, when really the time from discharge to age 5 when they go to school is much greater in the home. And we wonder whether or not this influence could be the microbiome as well. So I wish I had exciting data to show you for this. Unfortunately, I do not, but I can show you our plan. We've been funded by the ECHO program, which is Environmental Influences on Child Health Outcomes. Our project is the microbiome as a potential mediator of socioeconomic disparities and preterm infant neurodevelopmental trajectories from NICU discharge to school age. Because the means by which poverty alters neurodevelopment are unknown and obviously multifactorial. But we know that the microbiome is influenced by our environment and in turn influences brain development. So the previous speaker spoke about some of the ways that socioeconomic status can affect the microbiome. For the child, it affects whether or not they can play outside, the likelihood that they were breastfed, the number of caregivers they have, how crowded their home is. So all of those things can affect the microbiome and are determined by the environment. And we hypothesize that the microbiome is a biologic effector of the influence of socioeconomic status and environment on neurodevelopment. So this is our study design. We recognize that at birth there is a certain risk of poor developmental outcome just based on gestational age. The baby then enters the neonatal intensive care unit, and there is the possibility of some morbidities there. So this is what we're calling Epoch 2. We're looking at NICU morbidities, we're looking at the early microbiome, we're looking at interaction with the mother during the hospital course and collecting a lot of microbiome data and clinical data there. At the time of discharge, we're doing one neurodevelopmental exam, and then they enter the home. And during that phase, we are looking at, again, serial microbiome samples from both infant and child. We're looking at milk samples. We are doing some geocoding and looking at the environment. We are also looking at, we're asking questionnaires and then doing serial neurodevelopmental exams, multiple time periods up until school age. And then we're measuring school readiness between ages 4 and 6. So we have some patients that we had looked at years ago who are school age now. So for them, we have 2 time points, the time in the nursery and their current time point. For the patients that we're enrolling newly, we'll have much more data, many more data time points to determine if there is a microbiome that is associated with disadvantage. The hope is that the impact of our study will not just be confined to preterm infants. We know that infancy is where this microbial patterning begins, but it has influence for a lifetime. And a key, I think, is that the microbiome is modifiable. So again, disadvantage or morbidity in the neonatal intensive care unit is multifactorial. And certainly there are many people looking at many aspects of that. But I think a key thing for the microbiome is that it is modifiable not just in general, but perhaps for an individual child in a time frame that's relevant to them. And that could be a significant intervention that we could provide. So I'd like to acknowledge the team. I've had several people work with me in my lab and very excited about our ECHO team, multidisciplinary from here at the University of Chicago, including members here, including Professor Heckman and Chase Corbin, experts from the Microbiome Institute, experts in large data, geocoding, care of preterm infants, and so we're very excited about this study. Thank you very much. Yes?
I noticed that in your animal study, TLR9 was upregulated in the, are you going to look at the viruses?
We would love to, and I've actually been talking to people. We have the samples. We currently don't have the expertise for looking at the viruses, but it's definitely something to look at along with the fungi and others, definitely. We haven't done it.
And the other one, I was surprised that the FOXP3 was elevated, which I would have expected to be lower.
So the development of the immune system in the preterm infant, there are elements that are sort of elevated, whereas in other conditions they would be lower. So that interaction is something that we're looking at as well. There are many aspects of the developing immune system that are upregulated early on, and that is sort of the challenge. How do they become normally downregulated with time? Yes?
Thank you for a really wonderful talk. The microbiome transplant experiments are really neat from the high growth and the low growth baby into the life. I know you said there's just some babies that no matter what you do, they won't grow. But do you know anything about the differences between, in particular, those 2 donors or just in general? And then we looked at, are they getting their mom's own milk? And we looked at the milk composition as a potential determinant of high growth.
So for both of these patients, actually, they were on formula. And so we do know a lot about that. And so when we increase calories, the highest we'll give them is 30 cal, but there are a lot of additives. The truth is, I don't actually think that's good. For a long time, all we were looking at is the scale. We were just trying to get them to put on more grams. I think increasingly we're realizing that head growth and linear growth, and it's not just more calories and more growth are important, but even with that simple metric, there are babies who we can't get to grow. And we sort of give them time and eventually it works out, but it is associated with health in ways that we don't completely understand. And I think to some extent that is why growth works for us as a health outcome, because it incorporates a lot of different things together, but it doesn't seem to be just the milk. Now, could we make it all go away if we could give everyone mother's milk? So that is an open question. I think it's interesting, just to get on my soapbox a little bit, I think it's interesting that people have tried to solve the human milk issue in neonatal intensive care units with donor milk, because I don't actually think that is the solution that makes the most sense, and it is somewhat costly. I think we would do a lot better if we would put the resources into lactation consultants and support for their mothers, so they would actually provide their own milk to the babies. At some level, it ends up being a resource question. But if I'm guessing where you're going with that, I think that could potentially be a very significant intervention. And that's sort of why I put up that slide comparing probiotics and antibiotics and breast milk. There's a lot of work trying to figure out what's the optimal probiotic, or what can we give to these babies. And I think it could, at least for our patient population, start with very simple things. If we could have every baby receive its own mother's milk, which isn't actually a simple thing. There's separation. A lot of these mothers are sick. They don't get to stay with their babies. The babies can't suckle. There's a lot of reasons why it's not actually simple. But that could be a very significant intervention, separate from others. Yes?
So I thought it was very interesting to see the microbiota composition, both in the infants that didn't get NEC and the ones that did. And I think your top one was Comomonas, which is just sort of a strange environmental organism, which I think highlights the fact that the premature babies often don't get an exposure to a normal microbiota. You also showed that probiotics could potentially reduce the risk of NEC. There are, of course, limited case reports that probiotics can actually lead to septicemia in very immunocompromised populations. So I wanted to know if you could just comment on what you think would be best practices regarding initial colonization and probiotics in premature infants.
So interestingly, most of the studies on probiotics and preterm infants have been specifically around NEC. So the primary outcome has been whether or not they've had NEC, and they have evaluated incidence of sepsis in those patients, and the studies haven't shown a difference. So there have been case reports, and currently, I don't actually know of even if we all wanted to give probiotics, I don't know of an approved product, because there have been so many issues with that. The most recent one that was part of the large study in Australia and several others, Abidophilus, had to be pulled off the market because of yeast contamination. So there isn't a good product. One of the difficulties is that all of those studies gave the probiotic from the beginning of life to some endpoint, 36 weeks, a very long time period, the same probiotic. And I think part of the issue there is that's actually not how it happens. That's not diversity either, right? It's one or two organisms at very high levels being given to a preterm infant and doesn't follow any sort of developmental pattern. So best practices, I would say mother's milk, limiting antibiotics, contact with the mother as much as possible. So we try to do kangaroo care. And sort of just being vigilant about how we're treating these babies. So for a long time, there was just fear of sepsis. We thought that the biggest risk to these babies is that they would die of sepsis, and so there's a lot of antibiotics given. The data I showed was specifically around early antibiotics. So antibiotics are commonly given for the first 48 hours of life. Whether or not we can come up with different algorithms around antibiotic stewardship, as you raised, so that we don't have to do that, but also later courses. And could we be selective about intervening? So I think one of the problems is there's no entry into when you give probiotics except being born preterm. So it's not only best practices going forward, but how do we understand when microbial pattern is perhaps not optimal? And maybe those patients could receive a probiotic or have some sort of intervention, but not all of them. Sort of moving perhaps to a more personalized medicine, even for these preterm infants.
So
Yes.
I know that all the preterm infants get fortifiers. Do you use the human milk-based fortifiers here, or is it in your thoughts on? So instead of being made from cow milk,
Right.
which is
Those-
informational, but
Sure. Those are exorbitantly expensive. So we do not, and it's a cost issue.
All right, well, maybe we should thank the speaker and let's eat some food.
Okay.